in different order. Got it. Okay, so here's a real example. Here are related protein sequences. And you can see that they look incredibly different by eye, but they fold into the same protein in these different organisms. And a central task in bioinformatics is assembling a multiple sequence alignment, or MSA, where each column corresponds to some common position in an unobserved ancestral sequence. And this is difficult for two reasons. First is that there's a combinatorial explosion of possible ways to align sequences. Because these sequences are different lengths, there's just lots of different possibilities for arrangements. And second, we don't have a ground truth because we don't know how these sequences evolved from each other. But the reason that multiple sequence alignment is such an essential task in bioinformatics is that they're used for all sorts of downstream applications. So one example is to build phylogenetic trees. Another is to identify and hypothesize about the function of highly conserved regions. So maybe if something's highly conserved through evolution, it has to do with the function. And the application that I'll be talking about today is contact and structure prediction. So on this slide, I'll tell you a little bit about the intuition of how multiple sequence alignments are useful for contact prediction, and I'll go into the details later. So in this protein, we would say amino acid I and amino acid J are in position, are in contact because these amino acids are nearby in the 3D fold of the protein. And this can also be represented on this pairwise distance matrix here, where the ijth value is the distance between the ith and the jth amino acid. So how does knowing a multiple sequence alignment help you predict context? The guiding idea is that if knowing what amino acid is present at position I gives you information about what amino acid might be present at position J, then it might be the case that there's some sort of structural constraint over time acting between positions I and J, and therefore they may be in contact in the three-dimensional fold. And this is formalized by fitting a Markov random field. This idea has been around in the literature due to many people, some of whom, well, I thought I had a citation here. Many people, so it goes by direct coupling analysis or gremlin. There's a lot of names for this general idea. Okay. So the status quo in the field is that multiple sequence alignment construction is a pre-processing step. First, you get your multiple sequence alignment, and then you do what you want with it. So in the world of machine learning, this would look like you take a multiple sequence alignment, you input into your method, you output your prediction. The issue with this is that there's no gold standard way to generate a multiple sequence alignment. There's a lot of different algorithms, and all the algorithms have different parameter choices. So this ambiguity led us to ask the question, well, will we see improvements if we learn our multiple sequence alignment with some guidance from the downstream task we want to use it for? So our goal is to build pipelines that look like this. We input unaligned sequences, we align them using some, what we're going to call a learned alignment module into a multiple sequence alignment, input that into our differentiable method that's going to make some prediction, and then we will back propagate and update both the method for, say, contact prediction as well as our alignment all in one go. This way, whatever our method is guiding the construction of the multiple sequence alignment. The challenge with this is that we're going to need a differentiable way to optimize over what is fundamentally a discrete object, a multiple sequence alignment. Okay, so for the remainder of this talk, I will first talk about what our differentiable alignment method is. Then I will provide two case studies. The first has to do with learning alignments and contacts together using a Markov Random Field based approach. And finally, I'll talk about a case study we did with learning alignments in the context of AlphaFold. Okay. So we're going to start with the Smith-Waterman algorithm, and we're going to make some changes so that we're able to learn an alignment. So I'm talked about multiple sequence alignment, but the way we're going to do multiple sequence alignment is to take a bunch of sequences and align them to a query sequence. So we're just doing a bunch of pairwise alignment, and then we're going to concatenate that. So that's why I'm shifting into talking about pairwise alignment. Okay, so how am I going to change pairwise alignment so that I can put it in a neural network? Well, I'm going to modify the classic Smith-Waterman algorithm. So Smith-Waterman algorithm finds the highest scoring alignment between a pair of two sequences. The input is two sequences, say x1 through x5 and y1 through y3, as well as a similarity matrix of match scores. So the value at the ijth entry here is some blossom score between the value or the amino acid x4 and the amino acid y3. Then this is input into Smith-Waterman dynamic programming, and that will give us the single alignment with the highest score, where the score is the sum of the similarity scores at match positions minus some penalties for gaps. And yes, this is typically done with a blossom matrix. Instead, we're going to change the formulation so that our match scores are not determined by this blossom lookup table and instead are going to be learned with a neural network. The idea here is that the neural network would be capable of learning a richer match function as opposed to blossom. Blossom is completely context independent. It's saying how often in general are, say, lysine and alanine going to substitute for each other. Whereas a neural network has the potential to learn contextual information. And maybe it's using alanine in one context because it is a small amino acid, and a neural network would be able to potentially learn that type of information. So we're going to train a neural network, and I won't tell you the objective function we're going to train it with because that's going to be dependent on which application. But the idea is to input two sequences into this neural network, and this neural network will output one vector per position. So this vector here, say, is corresponds to the amino acid X4 in the X sequence, say it's alanine. So it's going to represent not just that this is an alanine, but rather this is an alanine in the context of whatever the surrounding amino acids are. Now we'll take our match scores by taking the pairwise dot product of these vectors. The idea is that if the amino acids are playing similar roles in two different sequences, then they're going to be embedded in similar parts of space. And so the dot product between those two amino acids in their context are similar. And then those vectors are pointing in the same direction. The dot product is going to be high. And now the idea is that with high match scores in these similar positions, this is going to encourage Smith-Waterman to align these positions. Okay, and the second change is that we're going to develop a smooth version of the Smith-Waterman algorithm that instead of outputting just the maximum scoring alignment is going to output a probability distribution over alignments. These two ideas are going to come together in our learned alignment module. So the function I was telling you about that will map from sequences to one vector per position is a convolution. So we're going to one-hot encode each sequence in our group of sequences that we want to align. And like I said before, one of them is going to be called the query, and we're going to align each sequence to the query. So they're all one-hot encoded, and then we apply the same convolution to each sequence. Now we have one vector per position that represents both what amino acid was at that position as well as what amino acids were at its neighbors. And then we'll take the pairwise dot product of these vectors. So sequence 1 with the query, sequence 2 with this query, and so on. And that'll give us a set of similarity matrices, which we then input into Smith-Waterman. And now we get a distribution over alignments, where this is a distribution over alignments of sequence 1 to the query, sequence 2 to the query, and so on. And just by concatenating this gives us a distribution over multiple sequence alignments. So this part of the pipeline of just using a neural network to learn match scores, we were after the most sort of basic architecture that you could think of just to see if this works and keep it really simple at first. The more interesting part mathematically was figuring out how do we go from this algorithm that is fundamentally an algorithm to optimize over a set of discrete objects and change it into a continuous version for a neural network. So this is where I'll focus most of my time in this section. So like I said, we want to be able to use this in a neural network. So we're after differentiability. So we need to make sure that our alignment function is differentiable so that we're then able to back propagate through it and learn the parameters of those convolutions that give us the match scores. And second, we want, which goes along with differentiability, we want smoothness. And intuitively, to me, this means that the network is allowed to consider multiple different MSAs at one time. And essentially, okay, one MSA is doing better for the downstream pass, shift its weight accordingly. So to summarize what this looks like, Smith-Waterman, if we're trying to align these two sequences, would say R and S definitely align to A and B, and U through X definitely align through C through F, whereas Smith-Waterman would be able to express something like this, like, yeah, R and S probably align to A and B, but U through F, maybe they align, or sorry, U through X, maybe they align through C through F, or maybe they align over here to W through Z. And to do so, we follow the approach of Blondell and Mensch in their ICML paper from 2019 regarding smoothing dynamic programming algorithms on directed acyclic graphs, or DAGs in general. So they have a really general formulation, and we took that and applied it in our case. So to explain how it works, I'll first talk about how we can think about the traditional Smith-Waterman algorithm as a problem on a DAG. So we can translate local alignment into a problem on a DAG by drawing this graph here, where each diagonal edge represents a match. So here this edge represents x1 matching to y1, or this edge represents x3 matching to y2. And each horizontal edge is going to represent a gap in the y sequence, and each vertical edge is going to represent a gap in the x sequence. Now, if you look at this long enough, it will be easy to convince yourself that there's a one-to-one correspondence between paths, ST paths in this DAG and local alignments of the two sequences. And moreover, we're going to weight the edges of this DAG so that the weight of a path is equal to the weight of the corresponding alignment. So in this example here, x1 aligns to y1. That's going to give us the score, the alignment score for x1 and y1. And then x2, there's a gap at x2, and then x3 is going to align to y2. So to compute the highest weighted graph, or equivalently the highest scoring alignment, we can use a standard dynamic programming approach. So the way this works is you compute the highest weighted path from S to vertex V for all the vertices V, and then you return the highest weighted path from S to vertex T. And so this is computed with a simple recursion. So the highest weighted path ending at this vertex is the maximum of the highest weighted path ending at this vertex plus the weight of this vertex plus the weight of this edge, or this vertex plus the weight of this edge. And this has a really nice interpretation. This function is the highest scoring local alignment of the first I characters of the X sequence and the first J characters of the Y sequence. This is just exactly what was on the previous slide. This is how it worked when we just wanted the highest scoring path. But now, like I said, we want a distribution over alignments. So instead of keeping track of this function FV at each vertex that has this really clear meaning of the highest weighted path from S to V, we're going to keep track of a smooth version of it. So we're going to compute a smooth version of the function f that's equal to the log sum exponential where this maximum was taken. So the idea now is, instead of having each vertex keep track of the highest weighted path ending there, it's going to keep track of a value that somehow indicates information about all the weights of the paths ending at that vertex. So now you should think of this as I have a DAG, and on the DAG, each vertex has a value, a function value. I can use these function values to define a distribution over paths from S to T according to this particular random walk. So the details of this expression aren't important. The point is that with these vertex values, I can use them in the edge weights to define a random walk over S and T. And if I do this in a clever way, following the work of Blondell and Mensch, a really nice property is true of this probability distribution over paths or equivalently alignments. So under this distribution, if you work out the details, the probability that xi and yj are aligned is exactly equal to the derivative of this score at this vertex t with respect to the match score of xi and xj. So what's important here is that this algorithm is, first of all, differentiable. Second, it produces a probability distribution over alignments that we can then use in our neural network. So to summarize, Smith-Waterman just gives us one alignment. We replace max with log sum exponential, and now we get a distribution over alignments. The code for this is available in our repo on GitHub, and we hope that if you're interested in learning alignments in the context of neural networks, that you look at our code. So we also implemented an affine gap penalty as well as a temperature parameter that I didn't talk about here. All right, So now we can use our code in the context of neural networks. So for the first time, we can input unaligned sequences, align them with this learned alignment module, which involves this differentiable Smith-Waterman, to output a multiple sequence alignment, or technically a distribution over multiple sequence alignments, input that into some differentiable method, say for contact prediction, output a prediction, pick our favorite loss function, and train both the method and the alignment at the same time. Okay, so of course, I hope at this point I've written this function down and you're convinced that it's differentiable. And so sure, you can put this function and learn with it. But the bigger question is, does it work? Is it going to be possible to learn alignments jointly with a downstream task? And this isn't clear to us like a priori, because I mean, typically how alignment is done is using this blossom matrix, which contains a ton of information about which amino acids are likely to substitute for which other amino acids. We're not using any of that. We're just saying, okay, if you input unaligned sequences and your goal is some downstream method, is that strong enough of a source of information for a neural network to then figure out what's an appropriate alignment? Okay, so I'll answer the question of is it possible in the next section, but I just want to pause, especially because this is remote. And so I don't know if I've lost you. Any questions at this time? Okay, I'll keep going then. Okay, so before I talk about the results, I'm going to be a little bit more specific about what I mean when I say we learn contacts in conjunction with alignments. So we looked at a neural network version of Gremlin for Markov Random Field based contact prediction. So what Gremlin does is it assigns an energy to every sequence in the multiple sequence alignment. And this energy function has two types of terms. Single site terms VI. So VI is a vector that tells us which amino acids are most likely to be present in column I. And these pairwise terms Wij, which tells which pairs of amino acids are most likely to be present in columns I and J together. And the idea is that we can think of this as a probability model where the probability of a sequence is proportional to its energy. So the way this model is parameterized is by finding matrices V and W that approximately maximize the probability of the observed sequences. The idea being that if you have a probability distribution that models your MSA really well, then it will be the case that these terms, these single site and pairwise terms, are really descriptive for your protein family. Then contacts are going to be predicted by normalizing the pairwise interaction terms Wij in this formulation. So I have a picture of that here. So here would be, you should pretend that each of these this is a 20 by 20 grid within this box here. And the magnitude, the average magnitude there is going to be indicative of how much pairwise correlation is there between columns I and J. And if there's a lot of pairwise correlation, then one might guess that there's some sort of contact and structural constraint between those columns. Oops. Okay, so this method really explicitly uses an alignment in order to assign an energy to a sequence. You need some sort of common indexing between the sequences, and that's what the alignment is for. So our goal is to do this Markov Random Field based prediction, but start with the unaligned sequences and learn both the alignment and the parameters of the Markov Random Field in one go. So that's what the objective is for our method SMURF, or Smooth Markov Unaligned Random Field. It's going to learn the alignment and the Markov Random Field parameters jointly, and it's going to use a masked language modeling objective. So the input is the query sequence, the sequence you want to predict the context of, as well as the set of related sequences that are unaligned. We'll mask out a random subset of the positions in the remaining sequences, and we'll feed this all into our learned alignment module, which is going to give us a probability distribution over alignments. Now, we also are going to, at the same time, be learning the parameters of the Markov Random Field. So here I've depicted the VI column specific terms and the Wij pairwise terms. So the way that we're going to predict which residue was masked in our first step, our mass language modeling step, is we're going to look at, okay, assuming a particular alignment, what is the expected energy of the sequence with the current Markov random field parameters, given all the different possibilities for what was masked. And then it will guess the amino acids that give the higher energy functions with greater probability. And this whole thing will be amortized over this distribution of alignments. And then our loss function is just how close, how accurate were our predictions in this mass language modeling task. And so our back propagation is going to both update the parameters of the Markov random field as well as the convolutions in our learned alignment module that produce the match scores that give us the alignment. But since this is a more technical audience, I will also mention something that was vital to get this to work. When we just ran this pipeline as was, we weren't seeing across the board improvements. We felt like it was hard for the network to find a good starting point. So what we ended up doing was some necessary pre-training of the convolutions that really helped give SMURF a good start to starting with a reasonable alignment. And then it was able to learn the Markov random field parameters and then at the same time adjust the alignment as it went. But just to get a reasonable alignment to begin with, we needed to do a step called basic align. And the input here was just the sequences, our query and the rest of them, input into the learned alignment module. And then we use this to get an average MSA. And we then computed a loss function between like how well does this MSA predict each of the sequences. In doing so, this incentivized the network to find a set of convolutions that produce alignments where each column is primarily one type of amino acid. So this is just like sort of a very cursory first pass to give our convolutions like some sensible, to be somewhat sensible. And then it was really in the training of the Markov random field with the alignment altogether that we are able to arrive at a much better, more refined alignment that was able to do contact prediction as well. Okay, so is it possible to learn multiple sequence alignments jointly with protein context? Okay, as I've definitely alluded to, yes. So our first question was like, can we just get reasonable alignments that give reasonable predictions of context? And we were really happy to see that not only this was the case, but on average, we saw improvement in the contact predictions. The improvement is mild, but we were still happy to see it because we were really just looking like, is this possible? So in this plot, each point corresponds to one of our one protein family in our test set. And the X value is a measure of how accurate is the contact prediction if you just use a standard MSA and put that into Gremlin, also trained with mass language modeling, but that alignment is fixed. And the y-axis is a measure of accuracy when you use mass language modeling to train Gremlin, but allow the alignment to vary. In other words, we used our method SMURF. So any point above this dotted line means that we saw improvement. So again, it's like very mild improvement overall, but if we plot the histogram of this difference in the scores, we can see here to the right of the line means that we see some improvement. Then without any sort of retuning of the model, we decided to try it on RNA. We found some pretty promising results that seemed like for some families, allowing the alignment to vary, enabled the network to find Markov random fields that produced pretty substantially better contact predictions. And so you can see the improvement here. I mean, it's a small number of samples, so it's still very preliminary, but we think it might be suggestive that there is more to be learned from realigning RNAs as compared to proteins. The vague thought is that proteins are somewhat easier to align because there are 20 amino acids, and if you find enough sort of heavily conserved motifs, you can sort of fill in the blanks. But with RNA and only 4 nucleotides, things are a little bit more difficult. So perhaps the RNA space is a space where improving alignment can help more. So we zoomed in on a couple examples. And so here's an example where our method SMURF did better. And so the red X's correspond to what SMURF predicted was the contact, and the blue was what the mass language modeling gremlin did. And so you can see here right off the bat, blue sort of has more points sort of scattered throughout, seemingly random, both in the example where we did better and the example where overall we did worse. So we kind of saw this pattern by eye and are interested to investigate more of why this is the case. The reason that this example did worse is there's a lot of false positives in these two particular areas. Okay, any questions before I move on to the final section of the talk where I'll talk about AlphaFold? Okay, I'll talk about our experiment with AlphaFold. So, as you guys know, AlphaFold is breakthrough in structure prediction, but it's not a single sequence model. It really uses evolutionary information from a multiple sequence alignment in order to make its structure prediction. So when we were working on this project, AlphaFold was open sourced and the results of CAS were out. And we had this thought, okay, well, maybe the few examples where AlphaFold did less well, it was because the input alignments were not as good. So we thought, okay, what if we use our method to learn alignments to optimize AlphaFold confidence? So we wanted to compare what happens when we input a set of unaligned sequences to our learned alignment module, which outputs a multiple sequence alignment. We put that into AlphaFold, that outputs a prediction. And now we're going to try to optimize the confidence of the PLDDT of the prediction and use this loss function of optimizing the confidence in order to update our alignment here. So we're going to backpropagate through AlphaFold. We are not retraining AlphaFold, not changing any of the parameters. We're just using the fact that AlphaFold is a differentiable function. And we'll compare that to what happens when you input the multiple sequence alignment produced in CollabFold through MMSeq2 to just run it through once and get a prediction. So to summarize the pipeline, input unaligned sequences to learned alignment module, get a multiple sequence alignment, put that into AlphaFold, and then optimize to increase the PLDDT, not changing anything about AlphaFold, just changing our alignment downstream. Since neural networks can be really finicky with random seeds and learning rates, we just repeated this process 180 times with different random seeds and a few different learning rates, and plotted the results over on this plot here. So this is for one protein family, and each point in this plot represents one time we did this training. And the value on the x-axis is the confidence of the AlphaFold prediction at the most confident it was in the trajectory of learning. And then the y-axis is the RMSD error for the prediction at that point. The blue dotted line is the confidence of the AlphaFold prediction from the MMSeq2 MSA, and the red dotted line is the error for the MMSeq2 MSA. So any point below the red dotted line means we came up with a structure prediction that was better than the one that was produced from MMSeq2. It shouldn't be surprising that all the points are to the right of this blue dotted line because we're optimizing for confidence. So of course, if we're optimizing for that, we're going to find solutions that are more confident. In an ideal world, we'd see a nice straight relationship where we'd have the most confident points have the most error and the least confident points have the highest error. We can definitely see that trend, but it's not as clear cut as one would hope. Okay, so this is what we did for 4 different examples from CAS where AlphaFold didn't do quite as well and the sequences were like reasonably linked. So for this example, we decided to look at the most confident point. So here corresponds to one training trajectory, and on that training trajectory, at what point was the AlphaFold prediction the most confident? And so the first thing we did was compare the structure predictions. So here in these examples, rainbow represents the true structure and gray is the predicted structure. So here we have the AlphaFold prediction from the M62 overlaid with our prediction. And here we have the AlphaFold prediction from our learned MSA overlaid with the true structure. And so it shouldn't be surprising that our structure is more in line with the truth as compared to this one. We sort of knew that already because we know that this structure has lower RMSD. So the second thing we did was compare alignments. Oh, okay, I guess I'll say one more thing, which is we repeated this for 4 examples, and then in 3 out of the 4, we found that if we looked at the most confident point, we and fed that MSA back into AlphaFold, we ended up with a better structure prediction. So if our metric is find better structure predictions by taking the most confident point, by that metric, we'd say that succeeded in 3 out of 4 cases. But we were really after the question of why. So our initial guess and what led us to look at this or even perform this experiment was, well, maybe the reason AlphaFold isn't doing well in these examples is because their multiple sequence alignments aren't as good. So maybe if we optimize them, we'll have better multiple sequence alignments. So as it can be really difficult to figure out what is a good multiple sequence alignment, what is a better multiple sequence alignment. Sure, it gives a better structure prediction, but is it better? So the first thing we did was look at them by eye, and luckily it's actually really easy. It's hard to say what's a good multiple sequence alignment, but it's quite easy to say what's a terrible multiple sequence alignment, and that's what we found. So here's the MM62 MSA for this family, and you can look at this clearly conserved motif here. It's almost entirely conserved across all of these sequences. And if we look at our learned MSA and what happens to this motif, which really should be in the same columns, you can see it's completely scattered throughout. There's also sorts of other inconsistencies, like 2 sequences in the MSA that look nearly identical to each other get aligned to the query completely differently. So we found that the multiple sequence alignments that yielded these improved structure predictions were low quality and inconsistent. And it wasn't just this example. We found this to be the case across the other examples where we were seeing improvements. So what gives? My takeaway from this is that AlphaFold is susceptible to the same quirks as other neural networks. So it reminded me of an experiment I heard about 5 or 10 years ago about ImageNet. So ImageNet is this neural network that classifies images, and people did this experiment where you input noise to the network, and then you say, okay, neural network, how should I change this noise just a little bit so that you're really confident that this noise is a banana, and change the noise a little bit in each iteration. And in doing so, what you'll end up with is a picture that looks like this. And I think we can all agree there's some banana-like components, but I think if I asked all of you to sit down and draw me a picture of a banana, no one would have drawn this. And you can see the same sort of thing happening with other prompts. And I think what we're seeing here is an analog of that. So likewise, aligning sequences in a way that's just trying to produce high confidence predictions from AlphaFold need not yield something that we think of as a reasonable alignment. I think this is especially important to point out for AlphaFold because, I mean, it's a great algorithm for predicting protein structure, as we know, but at the end of the day, it is a neural network, and you're going to see weird, unexpected behavior, especially if you try to do things like this that it was not designed for. Okay, so to summarize, we developed a differentiable alignment method by formulating a probabilistic version of a classic dynamic programming algorithm. The first thing we did was establish that it's possible to learn things. A downstream task such as contact prediction gives enough information to the neural network to arrive at a multiple sequence alignment that is then able to improve the contact prediction mildly and looks reasonable. I didn't put a slide up about it, but we definitely did a sanity check of the MSAs learned with the contact prediction and those passed. Right. And the second thing we did with our differentiable alignment method is we used it with AlphaFold to reveal unexpected behavior. That is that we can find these really weird, inconsistent, low quality MSAs that somehow improve structure predictions for particular families. There's a lot of places we could go in this differentiable alignment space. So first is that we could change the objective function to learn more reasonable alignments to improve AlphaFold structure predictions. So right now I was like, give me any alignment you want, as long as that confidence of the AlphaFold structure prediction increases. Perhaps there's a better way to change the objective function that also encodes some notion of like, it must be a reasonable alignment. I think this would be particularly interesting in light of the most recent CAFs, where to summarize some of the results is that methods that ran AlphaFold on an array of MSAs and took the best results did really well. So I think it is the case that AlphaFold is sensitive to the input multiple sequence alignment. So if we can learn better ways to align sequences, we're going to be able to better use AlphaFold. The second is to apply this alignment algorithm in other bioinformatics tasks that require alignment. So I'm going to point to some current ongoing work of mine with a great group of co-authors. We're currently using our code to train structure comparison methods. And so, my citation for it should be next. Okay, right there. Here's MLSV workshop link for another application of this differentiable Smith-Waterman code. And then finally, zooming out, this is one example of trying to figure out a way to optimize over a fundamentally discrete space, a space of alignments, and looking for ways to do that in the context of a neural network, which requires continuity for the differentiability. So another future direction is to design differentiable methods for other discrete optimization tasks. So the first thing that comes to mind is we did multiple sequence alignment in this really naive way where we had a first sequence and aligned each sequence to the first sequence. But can we do this in a more subtle way by doing something more hierarchical that builds more tree-like? You can also imagine trying to build trees in the context of a neural network. Really, there's a lot of opportunity in this space. So I'll end there. Thank you all for your attention. I'd be happy to take questions, and I'd also like to thank my funding sources and Harvard, where I currently sit, as well as my co-authors who are listed on the first slide.
Hi, I had a question. I was kind of curious if you looked at the alignments that you generated using like the contact prediction as the objective function, and whether those, like how those compared to the MSAs that you saw for using AlphaFold as the objective function.
Yeah, so once we realized how ridiculous the AlphaFold ones were, we definitely looked at the other ones, and they were much more reasonable. So we looked at a couple metrics. So the metric that I think we ended up putting in the paper was looking at pairwise consistency. So if you look at what's the blossom score of two sequences pulled out from the alignment in our alignment versus the input alignment, and see how similar those were. And so you could see that things were similar in that regard. But yeah, we didn't see any of this like really crazy, like there's a million gaps throughout.
Yeah.
Nothing like that.
The other thing I was curious about is just like what the kind of the statistics of the sequences that you were putting in here, like, you know, what was the sequence identity, but, you know, kind of between all the sequences that were going in, and what was like the length comparison, like were they all of comparable length and things like that?
Yeah, so the results in our test set was everything was between 256 and 512 in length. And then like deep in the appendix of our paper, we ran the method again on sequences of length less than 128, and we were able to see like really comparable results. So we don't think it's too length dependent. And then in terms of sequence identity, I think it varied family by family. But I think at some point we plotted sequence identity versus performance, and we didn't see anything that jumped out at us.
Okay, great. Thank you. I mean, really exciting work. I think this is really cool. Thank you.
Hi, Samantha, this is Daniel, and I have a question for you. Actually piggybacking on what you just said about different families. So some families will have more homologs, some other families will have fewer homologs. Do you see a difference in trying to model MSAs from families that don't have a lot of homologs out there and families that have a lot of homologs?
Okay, I just realized I think I answered the last question wrong. I was thinking of, instead of the length of the sequence, the number of homologs. So yes, the more homologs you have, I guess not necessarily the more homologs, having a lot of homologs that are diverse is helpful for this type of method because you're able to extract information over evolutionary time. And so most of the families had most of the families, when we ran our method, we downsampled, so they only would have up to 512 sequences. And then after, we're like, can we even do this with families that are much smaller, like 128 sequences? And that's what I was thinking of when I got confused and said it was the length.
Oh, I see.
It was the depth. I'm gonna revise my answer to the other question. Is I don't remember if the length mattered. I know we did some selecting of the length just for memory reasons. Really long sequences take a lot of memory, so we didn't use them. But it's the number of sequences that we were looking into, and we were pleasantly surprised that we were seeing like pretty decent results on our small set of homologs where the MSA depth was only 128.
All right, thank you. And this is really great work. I really like your presentation, and it's interesting. Your results with RNA are very exciting. I'll definitely follow
definitely want to
that.
spend more time in that space.
Thank you.
Hi, thank you for the great talk. I have a question about the RNA results in the second part. So did you use the seed alignment or the full alignments from the RFM?
I would like to look that up in the appendix of the paper before saying so.
I mean,
my guess is the seed, because those are more confident in that, but I should look at the details before.
Yeah, I mean, the way RFM works, if I remember correctly, is that you have known the consensus secondary structure beforehand, and you have this high quantity seed alignment, then you can just build on that using kind of a profile hidden Markov model. I think it's like some kind of stochastic kind of free grammar. Then you search more alignment into a big full alignments. So I suppose if you can use your method on the full alignment, that would be really awesome. But my question is about comparison. Did you compare to Inferno, which is a tool that built this RFM? And
no, we just did that very limited search of those 16 and compared to the existing alignment.
I mean, one thing I'm kind of worried about is that it seems your method can only kind of work on a set of online sequences, but with some kind of inherent consensus secondary structure. But with AlphaFold, it's a totally different task, because AlphaFold searches its multiple sequence alignment from a protein database, a protein sequence database, without any knowledge of the consensus, without any knowledge of the conserved structure beforehand. But RNA is totally different. So maybe you should use a similar fashion, not use the multiple sequence element from RFM, but search maybe in the NCB nucleotide database, and how well does your method will work in that case.
Yeah, we just took the set of sequences as like given from RFM. We didn't do any sort of searching with this, but I imagine we found in sort of this process with AlphaFold, it's very sensitive, the addition of homologs or sequences that maybe shouldn't be there. So yeah, the choice of sequences is probably very important and something that deserves more looking into.
Right, and one last question. So did you have to train for each alpha multiple sequence alignment, or can you train a single model that can generalize this across different RNA families?
Oh yeah, so that's a good question. So something that we did is the same for RNA and DNA, or sorry, RNA and protein, is that we trained our models separately for each family. So each family learned a separate set of convolutions, and something we're very interested in is learning a set of like what we think of as universal convolutions. So you shouldn't have to learn new convolutions for each family. Maybe learn one set of convolutions and you can apply that across different families and cut down on the
maybe we could try
time,
some kind of pre-trained language models, right? In the convolution can replace that with some kind of pre-trained language model, then use that to build a multiple sequence alignment. Seems very
yeah, well, we
natural to me.
wanted to learn from scratch, so that's why we started with sort of the basics of like let's do convolutions. But we could have learned the convolutions across families. Like very preliminary results were showing that's like a little bit harder. So in spirit of sort of can this work? Is the downstream fast enough information? We decided to make them family specific. It's a pretty tall order to generalize across the protein space like that, or RNA space. So it made sense that we were seeing better results doing family specific. It's a bit easier task, but it would be very cool to have convolutions that worked across protein space, or play with some of the pre-trained models to see, building on top of that. If we could build something on top of that, that would work nicely.
Right. Thank you very much. Thank you.
We also have a question in the chat, Sam, if you want.
Okay, let's see. Given that LAM outputs a soft MSA, does this mean that the unmasked residues in the MLL step are not one-hot encoded, but mixed residues? So the guess that we make is not like I'm guessing alanine, it's guessing a distribution. And then we'll compare that to a truth, which is just a one-hot vector. So the softness in the guess comes from two things. First is that it's a soft alignment. And the second is the fact that we're not guessing the amino acid with the highest energy, but we're guessing in proportion to the energy of the different potential amino acids there. Did that address the question? I'll say interrupt if it did not. Okay, let's see. I'm going to read the next one. Not sure I missed it, but I was wondering if you have or know of any current or future research on how to measure or optimize reasonable alignment from a human point of view. I would love the answer to this question. So yeah, what's a reasonable alignment? That's a hard question. So I still claim that the ones from contact prediction were reasonable. So yeah, that there's sort of consistency checks you can do, like if A and B are aligning to each other similarly, if you were to pairwise align them, is that similar to what you get if you pulled them out of the alignment you're getting? Like if there's a pairwise alignment of A and B that has a really high blossom score, and then you pull out from your alignment, what is the pairwise alignment of A and B look like? And it's like a really low blossom score. That seems silly. Like there's a better way to do A and B. And so like the optimal pairwise alignment of A and B is to sort of fit into the whole alignment. So that was sort of the point of view that we took to look at it, but I think it can be a little difficult sort of in general to figure out sort of what is a good metric. So I'd be curious to hear others' thoughts on that too. Okay, so return to the question, what about the unmasked residues? Yeah, so the unmasked residues are, so the unmasked residues are one hot encoded, and then we have a distribution of our alignments. And so like, let's just fix an alignment in our head for a second. And now you can easily compute what are the different possible energies of all the different amino acids at your mask position. And then, take the softmax, and then that would be your guess. And so that guess vector, we're just amortizing it over all the different alignments. So it was already a guess that was soft, and then we're amortizing it over different alignments. And so then it will remain soft. Okay, in the absence of any other questions, I'll make one more plug for our code. It's on GitHub, and feel free to make any comments or issues. We really are hoping that people are going to be using our code in other neural network pipelines to see if like learning the alignment at the same time helps. I hope that this presentation was a good case study that it definitely has the potential to help, but that you have to be very careful that you understand what your downstream task is, because if you put it into a black box, weird things might happen.
Okay, cool. Thanks, Sam. We'll have the recording available in a couple of days for anyone interested again, and hope to see everyone in 2 weeks.
Great, thanks.